Hey guys, we have just a teeny tiny bit of 11.1 left here, and then we're going to do 11.2 today. So we have this idea called replication. And we talked about in the cell cycle chapter about that DNA has to be copied during interphase so that then when the cell divides in mitosis, each cell has its own copy. But how does that copying take place? And this is the very, very most simplified version of that. But this is a process. DNA makes a copy of itself. Remember, it's during interphase. So it's before the cell is really starting to divide. And it occurs where the DNA is. So where's the DNA? It's in the nucleus. All right, so remember, the instructions of the DNA is on the inside. So if we kind of look down at the picture, all of the instructions that we need to copy are on the inside. So before we can copy in the instructions, we have to get access to the inside of the DNA. So that's the first thing we have to do, is get our two strands separated. And the enzyme that does this, right there in the picture, it's called helicase. Like hela for the helix, it's going to undo the helix. Okay. Next, new nucleotides are going to be attached. So I have a T that's now exposed here, so I'm going to put an A to that. Remember, our base pairing rules we're going to still follow. So apples in the tree, cars in the garage. So here this strand was all exposed, and we're going to attach these new nucleotides. There was a T there, so I'm going to attach one with the base of an A, because apples and trees, A's and T's go together. But then we can't just attach them. We have to also build these attachments, attaching one nucleotide to the next nucleotide. So that is an enzyme called DNA polymerase. Like remember, a polymer was something made of lots of things stuck together. So it is making the polymer DNA, right? It's actually doing the building of the new DNA. So helicase pulls our two strands apart, and then DNA polymerase builds DNA. Attaches new nucleotides together to form the backbone of the new strand. So these would be, remember, those strong covalent bonds that would hold this together. And then, full admission, I started making a video and I realized I started on the wrong page. So this is just kind of, remember, if you need to go over this, if you want to see the animations again, if you just go to YouTube and search Amoeba Sisters Protein Synthesis, then you won't have to do the Edpuzzle where you can't fast forward or, back up easily. So we have genes. And this is not new. These are segments of DNA with instructions. What we would have said, a certain trait when we were talking about genetics. But now we're going to say a certain protein. So remember, a protein is something we can use to build stuff, to make reactions go. So proteins are actually what cause traits, okay? And so remember, genes are in DNA. DNA is instructions. It can't do anything by itself. It needs RNA to actually get the job done. So we have three types of RNA. We have messenger RNA, or you can just write mRNA, and it's just a copy of the gene. So the DNA is in the nucleus. The mRNA is just kind of write down what is that instruction for that particular protein, okay? So it's the message. It's going to deliver the instructions from the nucleus where the DNA is out to the ribosome. Then we have ribosomal RNA, or rRNA, which is literally a structural building block of the ribosome. Okay, so when we're talking about that, we'll just say that's basically what the ribosome is built out of. And then we have transfer RNA, or tRNA, and it is transferring amino acids. which remember are the building block of protein. So the ribosome is where the proteins are built. Messenger RNA brings the instructions, and transfer RNA brings our raw material for proteins, our amino acids. All right, so step 1, transcription. I liked in the video that she said transcription before translation. You can remember C before L in the alphabet. So just like in replication, first thing we got to do is unzip that DNA so we can read the instructions that are on the inside. Next, mRNA is built using the rules of base pairing. Right? A's with T's, C's with G's, except, remember, if we're building RNA, it's always going to be, instead everywhere we would want to put a T, put a U instead. Okay? And then this was not in the video. This is a little bit of an extra thing that I think is worth putting in there. mRNA is processed. Okay, there's a lot of DNA that is kind of extra instructions. And if you've ever built a complicated piece of furniture, this would kind of be like the steps that you skip, or the steps that are in another language, the things that you're just like, I don't need to read those. And so before we go out to the ribosome, we kind of want to cut that extra stuff out. Okay, so we have what I'm going to call junk DNA, called introns. And the actual instructions are the exons. And easiest way for me to remember this is I think introns are in the way and exons are expressed. Okay, so the exons are what we want to keep. Introns are what we want to cut out. And so after it is written down following the rules of base pairing, chop out the extra introns, then it's ready to leave the nucleus and head out to the ribosome to actually build a new protein. So this is just a picture of, let's maybe label this picture transcription. Okay, so here's our DNA. All right, we had to unzip it a little bit, and here's the RNA. I'm going to put an M on there. It's like the mRNA getting built here. Okay, so this is going to go get built. So if this was our DNA, the blue is the DNA, it got pulled apart. And here we're building our RNA. T would go with an A. A will go not with a T, but with a U. C with a G. T still goes with an A, but again, everywhere we would want to put a T, we put a U instead. All right, so mRNA processing. If I was going to make in mRNA, I'm going to use 2 colors. So here it would be like a fresh string of mRNA just produced off of the gene. Okay. So remember I said we have kind of extra instructions we want to cut out. Okay, so we're going to go through that mRNA processing. And we're going to chop out the ones that don't carry instructions that we want. Okay, so we try to remember which ones were in the way that we need to get rid of. Introns. So after we process, we're going to take those 2 exons and put them together, and this is a mature mRNA. Okay, all the junk has been cut out. And those introns, they're just recycled. They just chop them back up to pieces, and we can use their nucleotides to build more RNA later on. All right, so then we have this concept of translation. So instructions for a protein are read by the ribosome, and our whole purpose of this, remember, is to build proteins, so that would be a chain of amino acids. All right, so first thing, the instructions are going to attach to the ribosome. What were our instructions called? They were the messenger RNA. So the message comes out, the instructions get to the ribosome, Before you start building, you need instructions, right? So the mRNA moves along the ribosome until we reach what's called a codon, which we're going to define in a second, for the amino acid methionine. Okay, and it's always the first amino acid. Okay, so sometimes they even call that the start codon, but you can also put methionine besides in the beginning, but it is always the beginning. So when we say codon, we mean a set of 3 bases on the mRNA. And hopefully when we look at the picture, that kind of helps make this make a little bit more sense. So codon is on the mRNA. All right, then we have our tRNA. Remember, our transfer RNA brings amino acids, and it has what's called an anticodon on the tRNA, and it's going to match up to the codon, and we're still following those rules of base pairing. Okay, so in RNA, A, U, C to G. Okay, we'll define that again. Anticodon is always a set of 3 bases that's going to match the codon. So the mRNA brought the instructions. The tRNA is bringing our building materials, the amino acids. What the heck is the ribosome actually do here? Okay, so the ribosome's job is to attach one amino acid to the next. Okay, so if we look down at my picture here, so these little upside down stocking things are the tRNAs, and they're coming in with these blue things that are my amino acids. And so then here's the instruction. So here's the codons. There's a codon. There's a codon. There's a codon. Here's my anticodons. And see, they match up. So the ribosome's job is actually to make these bonds, okay, to attach the amino acids together. So this continues until the mRNA codon says stop. Okay, there's actually 4 codons who don't code for amino acid, and they're called stop codons. Wait, that's not what we want there. We're going to talk about stop codons later. So the codon says stop, and basically, so there'd be some codon that doesn't code for amino acid, and that tells the ribosome, okay, that's the end, and it just lets the chain of amino acids go. And so our chain of amino acids right now is more properly called a polypeptide. Remember, peptide bonds are the bonds that hold amino acids together. And if it gets folded and, properly treated, it can become a protein, but it's not quite there yet. We can think of a polypeptide kind of like a baby protein. Let me know if you have any questions about protein synthesis, which is taking DNA and actually the RNA, taking that instructions and building proteins. Step 1, transcription. Step 2, translation. See you later.